So I don't know how many of you were affected by what happened after class on Friday. Just wanted to remind you that there are some resources on campus if people are still concerned about that. Posted them on D2L, and if you do need any particular help, I can try and do my best, although there are definitely professionals who should be better at doing that than me. So today we'll talk about poxviruses, then we get to the part of the term where we talk about the really cool, fascinating viruses, i.e. retroviruses on Friday, giant viruses next Monday, viruses of archaea next Wednesday, and I was not able to convince David Goodman to give that lecture, so it'll be me. And then we'll have a review on Friday, and then the term's over. How did that happen? Good question, although I can't say I'm too terribly depressed about that prospect, to be perfectly honest. So let's move right along. Last time that I lectured, talked about herpes viruses, and the herpes viruses that we mostly talked about. There are many different herpes viruses. HSV1 is the cold sore, the classic one that's been best studied and mostly what I lectured about. Genome is quite interesting, got the circular genome, but these repeated sequences, they keep getting inverted all the time, probably because of the recombination patterns that happen during replication. And everyone who's in our mutant viruses from hell lab looks like we've got some other weird thing going on with the repeated sequences as well. So interesting stuff there. Big difference in terms of structure, of course, is the tegument, which is that part of the virion which is outside the capsid but inside the membrane. And lots of proteins there, particularly the VP16 protein, which is the transcriptional activator, very important for activation of lots of herpes virus genes. Like almost all DNA viruses, and of course today is going to be that big exception, these viruses have to replicate their genome in the nucleus, so it has to get there, and it's all the transport process that does that. We've got immediate, early, and late genes, which is a good introduction, again, to what we're going to be talking about today. These immediate, early proteins are the ones which are going to be regulating the early proteins. Those early proteins are going to be regulating the late proteins. And also, those immediate, early ones are the ones that are most important for regulating cellular defense mechanisms. We didn't talk too much about the replication processes of these herpes viruses, but basically they've got pretty much everything they need to replicate their genomes, but still have to get themselves into the nucleus. The change to rolling circle is rather similar to one of our favorite bacteriophages that we've talked about in terms of making copies of the genome. And then the other take-home message with these herpes viruses, there's lots and lots of similarities to bacteriophages. Packaging is, of course, one of those. We talked really briefly about latent infections. This is what's least well understood about these herpes viruses, and the whole induction process clearly has to do with latent transcripts which are being expressed during latent infections, but how exactly those regulate latency, in particular induction, which of course is what people are very interested in terms of cold sores reappearing, but also for shingles, which is the chicken pox, which of course is not a pox virus, it's a herpes virus, that then will reappear. Any more questions on herpes viruses? I know it was like forever ago that we talked about these. No, not immediately. Then George talked about parvoviruses and some of the most fascinating viruses around, these cruciviruses or hybrid viruses. The parvoviruses are mostly understood as the dependoviruses, so really satellite viruses that are dependent on the infection of another virus, in this case the adeno-associated virus, and I didn't bring the nice prickly structure with me. Thought about doing so. One of the really neat things about these guys, of course, is their replication, because they've got DNA genomes with secondary structures present at their ends, which provide templates and primers for replication. And as we'll see, that's probably also how the poxviruses replicate. And the poxviruses, it turns out, have some of the largest double-stranded DNA genomes. The parvoviruses have some of the smallest, now single, quote-unquote, single-stranded DNA genome. Why I say quote-unquote? I didn't bring my list because I didn't make a new one. Why quote-unquote single-stranded? Instead, I'm going to put some in quotes. What does it mean? Yeah, because part of it is double-stranded. Now, yes, single-stranded, but I would always say single molecule rather than single-stranded in a case like this, because you have all these secondary structures. And anytime any virologist, or for that matter, anyone talks about a single-stranded virus of any kind, or really for that matter, single-stranded nucleotides, there's always going to be some kind of secondary structure where these things interact with each other. So that's the big message with replication. But a lot of the similar kinds of things are true with these circular, single-stranded DNA viruses, particularly this hairpin loop structure present in these single-stranded, again, quote-unquote, DNA viruses, which seems to be absolutely critical for replication. These are some of the smallest viral genomes known. Basically only require 2 genes, a replication initiation protein and a capsid protein. Some of them have more of these extra genes, but we're really not quite sure what they're for. And the replication here depends on a endonuclease ligase activity of the rep protein, and then, of course, the capsid protein encapsulated in the genome. And the really fascinating thing about the cruciviruses, which is what George is studying, and Dr. Nacho, who gave the other presentation, is that these capsid protein genes look like RNA virus genes. So somehow 2 viruses got together, magic happened, love happens, whatever you want to call it, and somehow they ended up recombining with each other. And this is a very strange occurrence, but as is true in biology, things happen and then we try and come up for an explanation of them. So that's where we stand on these single-stranded DNA viruses. Questions on George's lecture? So he didn't have any clicker questions, so I'll have one. Turned on. Start. Five. Given the responses up here, I think we want to talk to our friends about this. Neighbors, enemies, whoever you would like. So go ahead. I know it was a long weekend, many hours ago.
Oh yeah, there was a little thing.
The thing that recruited the thing to do the thing. Yeah. Yeah, I like that. Okay, let's go again. On our marks, get set, go.
Remember that little protein you showed though that came around and the special little protein you grew it in himself?
That's it, instinct. Instinct. Five. Vote early, vote often. Okay, where are we? Show results. Okay, we seem to be divided here.
Oh boy.
More or less half and half between RNA made by a cellular RNA polymerase and DNA made by a cellular DNA polymerase. What are the key aspects of this question? First one, what the heck is AAV2? Adeno-associated virus 2. Okay, so that tells us immediately that what kind of virus is this? So, or a parvovirus. And in fact, a dependovirus because it's adeno-associated. Okay, so single-stranded DNA virus. That's the first step. Okay, so what's the primer that is used for these particular viruses, which I literally just mentioned? It's a secondary structure, which is a hairpin, which is what? DNA or RNA? It's DNA. Okay, so now we have DNA made by a viral DNA polymerase or a cellular DNA polymerase. So do these things even have viral DNA polymerases? No. So what are we left with? E. Make sense? Not make sense? Yes, no. Some people happy, some people less happy. Yeah, in the back. What about replication at the ends? So replication at the ends, the way that happens, because it can reform that secondary structure. So it will replicate all the way out to the end, and literally the polymerase will fall off. But then that can rearrange to make a secondary structure which can then replicate through the rest of the genome. You do have to have the nick by the rep protein, which allows you to get to the other end of the genome, but that is not a primer. That is just making the primer, which is still the DNA, which is all made by the cellular DNA polymerase. Make sense? Okay. Okay, let's hide these. So now, completely opposite end of the spectrum, the poxviruses. These are double-stranded DNA viruses, and again, I have to put these in quotes again, and linear genomes. As we'll see, these guys also have secondary structures at the ends of their genome, which is also very important for their replication processes. They're larger and more complicated than herpes viruses. The genomes are bigger, they make more proteins, and the really big difference here compared to all of the other DNA viruses that we've talked about, and actually that we will talk about, even the giant DNA viruses are a little different here, is that these replicate in the cytoplasm. They don't have to go to the nucleus. And if they don't have to go to the nucleus, that means that they've got all the proteins that they need either encoded in the genome or in the virion to replicate a DNA, which is really pretty amazing. And so a lot of people think of these as kind of like mini cells or protocells that can replicate DNA in the cytoplasm. Of course, one of the reasons we know a lot about poxviruses has to do with human disease. Probably the reason that most of the faces in this room are white has to do with poxviruses, because eliminated a large portion of the indigenous peoples in the U.S. and in the Americas, I should say. We'll talk about vaccines, which is why we don't have to worry so much about pox diseases anymore. And then these pox viruses are also really good models for messenger RNA production, making large amounts of messenger RNA, even though the mechanisms themselves are really quite different. And we'll talk about some of those as well. Couple of key concepts as far as this lecture is concerned. I've been beating vaccination over the head a lot this term. I'm going to do even more today. So vaccinate, 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 and that is why we don't need to worry about smallpox anymore, at least not in terms of widely currently circulating. And we'll look at a little video here about the elimination of smallpox, which, as far as I'm concerned, is really one of the major medical public health accomplishments that humans have ever managed to do. Genome ends, also hairpin ends, but different than the parvoviruses, however, are probably replicating in a very similar way. One big difference relative to the herpes viruses, you remember a lot of the herpes virus genes are spliced here. There's no splicing whatsoever. Why is that? Because where does splicing normally take place? In the nucleus. Where is this replicating? Not in the nucleus. And then we also have these mature and enveloped virions, very strange structures that you end up with these virions. This is from Viral Zone. Again, a nice place to go and look for more details on some of these viruses. Sometimes they have one membrane, sometimes they have two membranes, and both of these are still pretty infectious. So we'll talk a little bit about viral disease. For actually most of the pox viruses, it's not a big deal. Smallpox, of course, was a big exception to that. What these genomes look like, they're big, long, double-stranded linear genomes. The replication, as it turns out, is pretty similar to AAV, or at least as far as we know. Transcription is really quite different indeed, and a lot of this has to do with the fact, again, that it's replicating in the cytoplasm, and so it's not using all of the cellular machinery for transcription. It's got its own machinery, which is similar in some ways and different in other ways. And then how the virion gets out has a lot to do with how you have these various different particles. The vast majority of what we'll talk about today, after we talk about viral disease, is Vaccinia, which people thought was the original vaccine used by Jenner, but actually probably isn't. Where, in fact, Vaccinia came from is an interesting question that no one has completely figured out yet. But smallpox, again, to remind people, viral disease was known long before viruses. There was just a paper that came out, I think it was in Nature, looking at hepatitis B viruses that people have isolated from really old samples, some, I think, 10,000-year-old human teeth. So a lot of these viruses have been around, as we know, for long periods of human evolution at the very least. And it was pretty clear that smallpox was around also from that period of time. The smallpox virus is variola. And as I mentioned before, probably why most of us have relatively white faces here, because it was brought over with the immigrants that weren't supposed to come, illegal, et cetera, who then came and said, oh, this is, we like this place, we're going to spread all of our disease here so everyone else here can die. Smallpox is, even now, has a very high fatality rate, 25% or so in adults, and much higher in young adults and babies. And the classic line before the vaccines is you really couldn't, count your kid as your kid until it was a couple of years old or had already survived smallpox. And that was a lot of the infant mortality that was happening in earlier times. So where does smallpox come from? As far as we can tell, only replicates in humans, which is probably one of the reasons we're actually able to get rid of it. As we saw, one other virus seems to be the case that we're getting really close to getting rid of is, which? Polio, exactly. There's also molluscum contagiosum, which is another pox virus that my kids have had, in fact, many kids have had, doesn't seem to be a big deal, and is self-curing. Vaccinia, I love this, unknown. So probably came from some of the original vaccine strains, but has since probably been replaced by others, and that's probably because this vaccine has been around for such a long time that it's gotten contaminated by other poxviruses, etc. There are a number of other poxviruses, monkeypox. There have been a few cases, particularly I think last year in Africa there was a monkeypox outbreak. Unfortunately, it's called monkeypox, even though the reservoir hosts are rodents. Cowpox, also in rodents. Fowlpox is a bird. Myxoma, I'm not going to talk too much about myxoma, but this is the way that the Australians were going to take care of all the rabbits. Unfortunately, the rabbits became resistant. And so, but again, basic message here is that there are lots of different pox viruses. It's not just smallpox. So why do we care about smallpox other than the fact that it killed lots and lots of people? It's because now we know a lot about vaccination. And so vaccination really came originally from variolation, and variola, of course, is the name of the virus. This was known in India and China well before Jenner came on the scene, and basically because they took good records, they noticed that someone who had been infected and recovered, which was still 60% of young adults, were now much less susceptible to future infections. And what they eventually found was they could take small amounts of virus and infect someone, and they would then be resistant to future infection. So basically, hit them with a small amount so they don't die, and then they're resistant later on. Even so, if you're actually using the variola itself, still about a 1% death rate from doing this. And I don't think any IRB board would accept a 1% death rate for protecting yourself against other diseases. But still, 1% is a heck of a lot better than 40%. So this was done really apparently quite commonly before Jenner in the late 1700s noticed that cowpox, which of course is a rodent disease, but it was the milkmaids that all had beautiful skin. Why do they have beautiful skin? Because they hadn't gotten pox, or at least not smallpox or variola. So why was that? Probably because they were exposed to cowpox. So let's see if we can protect people from variola by infecting them with with cowpox. And so the idea was you use this cowpox virus to protect against smallpox. And this worked incredibly well. And eventually, Vaccinia was developed from this. And Vaccinia and vaccination at all comes from cowpox. So the original vaccines are from the vaca, which is interesting because apparently Pasteur, when he came up with his vaccines for rabies a little later on, they thought about trying to change the name and said, oh no, we'll just stick with vaccination. It works. So again, we have Vaccinia now, and because of Vaccinia, we've almost completely eliminated smallpox. There are two, as far as we know, stocks of smallpox, one in Atlanta and the other one in, if I remember correctly, Novosibirsk in Russia, all particularly well contained. And there's been a lot of discussion about whether people want to simultaneously put their last stocks of smallpox in the autoclave simultaneously in these two places and hit go and get rid of it completely. There's actually an advisory committee on variola research, and this guy right here is one of the members of the WHO advisory committee, on variola research. And here I am with Dennis Bamford, who has been very involved in virus structure determination, particularly the really old viruses, the double jelly roll. That was kind of his theory that all of these viruses arose at a very old time. And this is the replica of a skull at a place called Atapuerca, which is just outside of Burgos, Spain. And in fact, they just showed that these are hominids from probably over 100,000 years ago. Where they have viruses associated with them, we're still waiting to see. But basically, I had a long drive with Antonio here coming back to Madrid from the meeting. And basically, their message is, we want to hang on to the actual virus stocks rather than completely getting rid of them. But the only thing that you can use these particular stocks for is for testing antiviral diseases. And basically, every year they meet, they say, okay, do we need to get rid of this? Should we not get rid of it? And then even more recently, some of you may have heard that horsepox was actually made in the lab. There's a biotech/academic group in Boston, if I remember correctly, who literally bought all of the pieces and put them together to make horsepox to try and make a vaccine, again, particularly against smallpox. There are some issues with the Vaccinia itself in terms of inoculation and some virus-related disease. So they wanted horsepox as a way to potentially study this, and they were actually able to make it from scratch. And so there's a lot of discussion about whether you really need to have the actual virus if you can make one from scratch. Now, you also might say, well, what's stopping people from setting up their own lab and putting all these pieces together for smallpox? Because the genome sequence is immediately available. Fortunately, most of the companies that do this have software that says, hey, you're making sequences that look like smallpox, and we're going to make all these red flags and say, hey, we're not going to sell this to you. But there's some interesting aspects about that and lots of interesting discussion that I'd be happy to have after the term at Rogue. So the vaccination, and again, we'll talk more about this a little later on. There's large amounts now of what I, again, like to call the pro-disease groups, but that's actually been around for a rather long time. So this was a pro-disease publication in 1812. This is supposed to be Mr. Jenner, and look what happens to poor people who get vaccinated. Cows start to spring out of their arms, or their buttocks, or wherever else, the nose. So this is obviously a horrible thing to do, is this whole vaccination business, because it'll make cows. Yeah?
So the difference between would that be like the dengue that has different stereotypes, or is it totally different?
Okay, so the question is here basically, what's the difference between the different pox viruses? And you might mean Vaccinia rather than cowpox, but that doesn't really matter. So yeah, and then relative to dengue, which has these different serotypes. The dengue viruses are very similar to each other in terms of their genome sequences, what their host ranges are, etc. On the other hand, the pox viruses are very different relative to each other. And in fact, it's pretty amazing that the Vaccinia virus actually does give protection against variola. And that's quite different than even for something that's very closely related as dengue. And quite why that is, I think nobody really has a good idea. So this was 1812. This was a couple of years ago, which all the wonderful things you can do with Photoshop. But my favorite graphic as far as this is concerned is this one, which you can get from that particular link here, but I think was originally put together by the CDC. Basically on the left-hand side here, annual morbidity in the US in terms of different diseases, diphtheria, Haemophilus influenza, hepatitis A, hepatitis B, measles, et cetera, et cetera, et cetera. And now after the vaccines have been introduced, the numbers here are basically zero. We went from 16,000 a year of polio to zero, 47,000 for rubella, smallpox almost 30,000 to zero. Unfortunately, this is 2011. I'm afraid some of these red bars may be actually getting a little bit bigger, which is rather frightening. You know, here's 61 cases of measles. I think there have been 61 cases of measles this year already. So take-home message, vaccinate, vaccinate, vaccinate. There's also really great, if you haven't seen this, John Oliver talking about vaccination, incredibly well done. And one of the comments I really liked on this YouTube, we're not going to take a look at this one, is that one of the commentators said, I really don't like to agree with Englishmen because I'm Scottish, but it's a really good one. So, so I did, however, want to show this brief video, which is about Donald Henderson, who passed away last year, the year before, I believe, in terms of actual elimination of smallpox. So it's a rather nice video, and if we have a chance on the last lecture, I'll show another segment from this particular one. There will be a clicker question on this, so you want to watch carefully.
Dr. Donald Henderson accomplished what no man has ever done before. He completely wiped out a virus. And not just any virus, but the deadliest and most feared of them all. Smallpox. The virus killed 300 million people in the 20th century alone. And it left millions more disfigured and often blind.
It's been said that smallpox has killed more people than all the wars in history combined.
Donald Henderson is still haunted by memories of visiting the victims.
Their faces were anxious, there were flies all around. Some of them were crying, some of them you could see were dying. The odor was just overwhelming. It was like rotting flesh. And the British physician I was with put his hands on the railing and he said, I could never do that again. I could never move around on these patients. That is the most horrible thing I've ever witnessed in my life.
The battle against smallpox, in fact the outright war on all infectious diseases, started in earnest at the end of World War II. It was an era of science and progress, a time of confidence, some would say, of hubris.
There was a tremendous optimism that science was going to protect us and it was going to solve all our problems. Penicillin had just made its debut, and this was effective against bacteria. It was a kind of sense that we would have something for viruses soon, if not immediately, and in any case, we had vaccines.
No one doubted germs would surrender to medical ingenuity, and they did.
BDT, the miracle insecticide that helped prevent epidemics in war-torn Europe and the Pacific areas, goes to work in an American community.
It seemed like a golden age when many of the diseases we used to fear in the past, like whooping cough, diphtheria, smallpox, polio, all seemed to recede into the woodwork because we could control them.
With smallpox and other infections apparently in retreat, research could focus on the remaining killers, chronic diseases like cancer and heart ailments. These were now the biggest challenge in the war on disease. In 1954, as a newly minted doctor, Donald Henderson was ready. It was a time of optimism. It was a time of delusion.
These were great triumphs of public health and great triumphs of science, having these vaccines, controlling these once feared and once deadly diseases. But what really happened was sort of more out of sight is out of mind. They were gone only in our thoughts.
Bradford faces a grim situation.
In reality, outside of North America, smallpox was very much alive.
In the early 1960s, there were many importations of smallpox into Europe. Part of the reason was there were big outbreaks in India and Pakistan, and they were coming across as visitors or immigrants and starting outbreaks. And I was concerned that it was going to happen in the U.S., and we hadn't had any smallpox for so many years.
The world's wealthy nations, where smallpox was a fading memory, now had to face a stark truth. They would never be safe until the virus was destroyed everywhere. In 1966, the World Health Organization selected Donald Henderson as the man to do it. He was under 40, old enough to lead, yet young enough to believe.
We were engaged, if you will, in a crusade to do something that had never been done before. It could be done.
When the global eradication program started, there was smallpox in 43 countries, 15 million cases a year. Vaccine was scarce, and there was never enough money. All the team had was heart.
They were young, they were willing to spend incredible amounts of time in the field, they worked very hard, and let's say that they lived from the field. They were not very good at embassy cocktail parties.
The virus had one fatal flaw. It existed only in humans. Since no animals carried it, if every human at risk could be vaccinated, smallpox could be beaten. In the first year of the program, Henderson's teams vaccinated over 25 million people on 3 continents.
Smallpox is by far the easiest of all the diseases to eradicate. You need to vaccinate only once and the individual's protected. You had a vaccine that was very stable. You knew where the smallpox was all the time because everybody who could transmit the disease had a rash, a very typical one. There were no silent carriers who could spread it.
But teams still had to go out and hunt it down. A photo of a baby with smallpox said everything people needed to know to help find the victims. Slowly, one needle at a time, the killer was cornered. By 1977, smallpox survived in just one country in the world, Somalia. And then in just one person. Ali Mao Melon, a 23-year-old hospital cook in America, was the last natural case of smallpox in the world.
It would ironically be a case that should never have happened. He was a young man who had been a vaccinator, but he never got himself vaccinated. And then he came down with smallpox. And by this time, he had contact. I think the count was about 110 people.
Now it was a race against time. A frantic vaccination campaign hit the streets. That one last case meant almost 55,000 people had to be vaccinated, and fast, to avoid disaster. Henderson and his team beat the odds.
It was the first time in all of recorded human history that people had actually, with all of the species we have driven to extinction, actually managed to drive an infection, an infectious disease, to extinction, intentionally.
2 years later, the world was officially declared smallpox-free.
The end of smallpox should not be viewed only as the closing of a magnificent chapter in international collaboration. It should also be seen as one of the launching platform for our goal for health over by the year 2000.
The year 2000.
How do you feel? I suppose that as people said, it's, you get to the top of the mountain. It's wonderful. But the fun was in the end. And I think we all realize that we spent a lot of time in the field, a lot of good friends, lived through a lot of bad times. And it was all over.
Donald Henderson's victory was a spectacular exception. Smallpox is still the only virus in the world that's been completely wiped out.
Not completely true.
The war on infectious diseases had really just begun, and the deadly new viruses would be much more stubborn.
Okay, so now we need to have our clicker question, as I promised you, right? Me? Who, me? It's my job. I keep going for that elusive 100%, you see? People are messing with me. It's only fair. I'm the one up here, so. Thank you. So again, I can see how people vote here. So someone was clearly pushing all the buttons on their clicker. And I could probably go out and find out who it is, but I won't go that far. Okay, so now let's talk about the interesting aspects of smallpox, having to do with what it looks like. And again, this is the really fascinating part about these particular viruses. Again, Vaccinia is not obviously a cow virus. It probably was originally a cow virus, but basically what happened is people took the lesions that were on anyone and then literally ground those up and used them as an injection. And in fact, one of the ways they got the vaccine from Britain to the US was actually send infected people on ships and then get their lesions here and then use those. So really amazing process. But that particular virus has literally hundreds of proteins packaged in the virion. So we've gone from just straight nucleic acid coming inside the cell. Now we've got massive numbers of proteins and a very large genome. Actually, it's the largest genome we've talked about so far, about 200,000 base pairs in length. And at either end of the genome have these inverted terminal repeats. Again, so you've got one sequence on one strand, the same sequence on the opposite strand, and these guys all base pair with each other. And what that means is that this sequence here will also base pair with this sequence over here at the other end. And at the ends of the genomes, there's about 4,000 bases that are non-coding, and that's probably because you have to have these inverted terminal repeat sequences which have to be conserved, because if they're not conserved, you're not going to be able to replicate properly. And that comes from looking here at these ends of the genome. Again, almost a 200,000 base pair genome. If you zoom into the ends, you have these repeated structures. You keep zooming in, you end up with these two strands, and so there's this sequence here, 5' to 3', 5' to 3', and then these loop structures where they're almost completely base paired all the way out to the very end of the genome, and the 5' is contiguous, just goes around the end and goes back to the 3'. And so, again, I have this in, quote, double-stranded. It's really a single molecule, which is almost completely complementary to itself. So depending on how you want to define this, you could call it double-stranded or single-stranded. Single-stranded and completely complementary because it's one molecule, or double-stranded with these covalently closed ends. Most people talk about this as double-stranded with covalently closed ends, but hopefully you see that there's a difference between these two. If you just look at the sequences, and now this is the very end of one of the genomes, so here going 5' to 3' would be TAG. That does absolutely perfectly match the sequence at the opposite end of the genome, which would be, again, the complement of this CTA. To be able to get around this corner, go from one strand to the other, they can't actually be base paired, because you can't be base paired and then just hop around. Just the structure of DNA isn't such that you can do that and go back in the opposite direction. So these very last ones at the end of the genome are actually not apparently paired with each other. So that's what the ends look like of these genomes. What is encoded in these genomes? Basically, absolutely everything that you need, with the exception of all the splicing machinery. So it's got DNA glycosylases, DNA primases, kappa enzymes, transcription factors, RNA polymerase subunits, decapping enzymes, etc. Another thing to notice here is you don't have a nice separation of early and late genes like we had with SV40 and those other viruses. You know, all the early genes are together, or the late genes together. Here they're all completely mixed up with each other in the rest of the genome. So, and we'll talk about how they're regulated here in just a second. What are they in? They're in these very large particles. Again, the largest virions that we've talked about so far, a couple hundred nanometers by about 100 nanometers, that have these very strange sort of hourglass-like structures in the middle. I kind of like to think about this like the viral nucleus, but also an envelope, and because they're poxvirologists, they call this a lateral body instead of a tegument, but basically it's still the same idea. Between your capsid on the inside and your membrane on the outside, you also have lots of proteins. There are two flavors, if we want to call it that, of infectious virions, as I mentioned right at the beginning. We've got those that have one membrane and those that have two membranes. The one with a single membrane, what's called the intracellular mature virus, and we'll take a look at that in a cartoon in just a second. And then you have the extracellular envelope virus, which has another membrane on the outside of this one. And so a extracellular envelope virus, the EEV, is an intracellular mature virus with an extra membrane around the outside of it. Now, why does the virus bother to do this? Again, why answers are always going to be evolution. These guys seem to be much better at getting from cell to cell inside one organism. These guys are much more stable, as they mentioned in the video. These then are really good at transferring from organism to organism. If we're talking about variola, it's going to be from human to human. And these are the ones that are most stable, and this may or may not be apocryphal, but one of the ways that people are said to have besieged towns in the Middle Ages is take someone infected with Poxvirus and dead and then fling them over the wall in order to infect the people who are actually there. And that's because of these very stable virions. What do these look like? We've got our intracellular mature virion, which can infect, an extracellular enveloped virion, which can also infect. Normal infection process here, coming again from the outside, because these are the stable ones, interact at the membrane or sometimes in endosomes. The core is released together with these lateral bodies, and again, just like tegument in herpes viruses, you have lots of proteins here which are involved in both shutting down cellular responses, but also in activating all of the early genes. So early genes, and in this case, these early genes also include the what are called intermediate gene transcription factors, so viral intermediate gene transcription factors. Those then are going to be important for replication of the genome, but also expression of these intermediate genes. These intermediate genes are going to be expressed once you have DNA replication. Again, all this is happening outside the nucleus, so all of these proteins are being made by viral machinery. Then the late proteins, which are going to be all of your structural proteins, are made from this replicated DNA. Among these late proteins are some of the proteins you're going to need way back over here at the very beginning of the infection process. Those then get packaged and can either be put into two membranes, where when this fuses with the plasma membrane, you'll end up with a single membrane bound. In some cases, these will pick up a third membrane, which will then fuse on the outside of the cell, and just two membranes that are present here when you leave the actual cell. And we'll take a look at that again in a cartoon in just a second here. But the amazing thing here is that all of the proteins you need for this first step are present in the virion. So what are these? First one is an RNA polymerase. This is a DNA-dependent RNA polymerase. Unlike bacteriophage T7, this has multiple subunits. The structure actually looks a lot like eukaryotic RNA polymerase. Where did it come from? Did our polymerase come from this polymerase? Did that polymerase come from our polymerases? Who knows? But again, a multi-subunit complicated polymerase has multiple proteins that are involved in stimulating transcription. It has proteins that can cap the ends of your messenger RNAs. It's got proteins that are important for transcriptional termination. It's got proteins that make polyadenylation. So again, everything you need to make messenger RNAs comes in with the virion. So what first thing you need to do, you need to transcribe. And so that's that process. Also a number of genes that are important for starting DNA replication, turns out are all part of this enzymes in the virion, including even a DNA topoisomerase that'll change the supercoiling state of your protein. So basically, all of these proteins are present in the virion when it comes inside the cell. The things that are not present in the virion are all of the DNA replication proteins, because you don't need those, because you can transcribe all of them from here. But what's encoded there are DNA polymerases, DNA processivity factors, again, more topoisomerases, single-stranded DNA binding protein, really everything that you need in order to undergo DNA replication as well. So you've got all of the machinery that you need for making messenger RNAs, you've got all the machinery you need for making DNA, but there's a problem. What's the problem? Now, energy is one thing, but what else? Not so much the right environment, but what do you need in terms of DNA polymerases? You need the nucleotides. Are there deoxyribonucleotides in the cytoplasm? No. What is in the cytoplasm? Ribonucleotides. So, having DUTPases and uracil DNA glycosylases, and also thymidine kinases, ribonucleotide reductases allows you to take RNA and convert it into DNA. So these are all viral proteins that'll take RNA, convert them into DNA. And most important of these is actually the ribonucleotide reductase, because it takes RNA and makes DNA out of it. So that's the process of making more of the DNA. Now, how are these genes regulated? You've got, again, ridiculously creatively named early transcription factors, intermediate transcription factors, and late transcription factors. Again, all TF, TF, TF. All of these, the intermediates and the late transcription factors, happen after replication. And of course, replication and transcription all occur in the cytoplasm. Okay, do we want to do another clicker question or want to skip it? Let's do it. Okay. Start. There we go. It's out to get me. That would be even meaner than I am. Okay, there's still a difference of opinion here, so let's talk it up briefly. Okay, be ready to go again? Let's do it.
Oh, I see. That's great.
It's not the point. Good practice for the final. Three. It's made late because you need it early, and that comes in with the virion. Too much stress for about, I guess, Wednesday mornings now. Sure. They'll also be made late. So all the structural proteins are made late. Okay, so very late and immediate early. Those are the two extra options that I had to put in because I wanted 5 answers. And not very many people like them, so I need to come up with better, incorrect answers, I guess. So the Poxvirus RNA polymerase, again, as I mentioned, is a multi-subunit polymerase that looks a lot like eukaryotic polymerases, and all of these viral early transcription factors, intermediate transcription, late transcription factors really function a lot like general transcription factors. So they're all about getting the polymerase, which is supposed to be this big blue thing here, to appropriate promoters, and these promoters are very different than any of the cellular promoters, which again makes sense because it's not even close to where the cellular DNA-dependent RNA polymerases are. One interesting aspect of these messenger RNAs is, yes, they get capped and tailed, but it turns out that the capping and tailing proteins are actually some of the same proteins. So guanylyl transferase and ribose methyltransferase, these are what you think about in terms of cellular capping enzymes, but it turns out they're also important for making poly A tails. Quite why that is, it's probably just economy of genes that are made in the genome. Yes, you have 200 kb, but the host has a heck of a lot larger genome. So these are proteins that are both used, again, at the 5' end and the 3' end of your messenger RNAs. Mentioned the genome already. This is what George talked about at the last lecture, these hairpin end structures which provide primers for replication. Probably exactly the same thing happens with poxvirus genomes. You have a nick that provides a 3'OH that extends to the end, and once it's extended to the end, again, this is self-complementary, so it can fold back on itself, provide a primer which can replicate all the way through the opposite end of the genome out to this end of the genome, This gives you a concatemer, as we've talked about before, end to end, 2 copies of the genome. So now you need to split these guys apart. There are lots of topoisomerases, as I mentioned before, in the viral genome. Those are probably really important for this process here at the end. Messenger RNA production, as I mentioned before. Termination poly-A tail are made by capping enzymes, so it's at the 3' end as well as the 5' end of your messenger RNA. Some of the late RNAs, strangely enough, get poly-A tails put on the 5' end of the genome. Or say not the genome, but of the messenger RNA. Probably important for transcriptional regulation and translational regulation, particularly for translation. Quite exactly what it's doing there is not entirely clear. Last thing that I wanted to mention is where these things are all being made. So again, this is a DNA virus that's replicating in the cytoplasm. If you look at a Poxvirus infected cell, unfortunately this is mostly Vaccinia virus infected cells, you see these regions of the genome separated from the nucleus where all of the other organelles are gone, and it just seems to be completely packed with viral proteins, viral nucleic acid, viral enzymes, viral activities, and in some cases this is almost like a different organelle inside the cell. And many people call these virus factories, where all of the virions are being put together, again, separated from the nucleus. And we'll see this also happens with some of the other giant viruses a little bit later on. Mentioned multiple times these immature, mature virions with one envelope on them. If they're going to become one of these enveloped virions, they pick up an extra envelope, which then will be released from the cell. Most importantly is that this extra envelope has actin polymerization proteins that are associated with it. So these Vaccinia can actually be projected from the surface of the cell into another cell just by the polymerization of actin. We don't have time to look at this little YouTube video, but it shows how these Vaccinia virions are literally pushed from one cell to another because of the polymerization of actin that happens due to the proteins in the membranes of these enveloped extracellular viruses. So on Friday we'll talk retroviruses. Ooh.